Well, I think I do know most people, but just in case, Rodney Ortega with Millipore Sigma Biocontrol. I've been here almost 20 years, I guess. So I've been working with GDS since the inception. What I was going to go through is just really two quick PDFs. One of them is what I think a lab should definitely have a really solid understanding on. The other one's a more in-depth PDF on what is actually causing the problems. I don't know if anyone really needs to understand why some things happen. If you guys want some troubleshooting as to, some really odd lines or, odd sample analysis, you can ask us and, me, and I can help out with the explanation as best as we can. We can't always determine, why something doesn't perform as it normally would, but we can at least give you what, our historical knowledge is on anything that doesn't look right. So with that being said, please don't hesitate to stop me at any time if I say something that you don't understand or doesn't make any sense. But what I did want to do is go ahead and share my screen so we can see what I wanted to show you. And this right here, this is a very basic GDS interpretation. I don't even know if I want to call it a guide. Really what it does is it shows you what the lab should see. I would say 95% or greater of the time when samples are positive. These are very typical positives. You know what? Let me actually, before I start there, let me go back one step. I want to look at, this is the true interpretation guide. This goes through a lot of different examples with explanations. As you see, this is a very typical guide with strong positives all the way to weak positives. But what I really wanted to go over here was this graph. This is pretty much a PCR graph that you'll see in any textbook that you read on DNA amplification, on real-time detection. So basically, as the amplification, the instrument starts, amplification will start. With GDS, we definitely have the threshold line that the sample line must cross for the sample to be positive, or if it's the internal control, it must cross that to be a valid negative. So what you see is DNA amplification occurring. These are the cycles. You don't see it listed here, but if you look at the GDS, you see number of cycles moving across horizontally. Once the DNA starts amplifying to a point where the system is detecting enough, you see an exponential jump in the line. The line will move in an upwards fashion like this. That is always true DNA amplification. When you look in textbooks, you always see this plateau phase. Basically, what's occurring here is within all the amplification tubes, there's a limited number of nucleotides, and primers and probes. So only so much double-stranded DNA is going to be able to be formed. When you see this plateau phase, there's essentially no more reagents. I don't like that as far as teaching that, because in GDS, if you notice, you don't see a plateau phase. If you look over here to the left, you see a normalized fluorescence. This is the fluorescence level. The plateau phase within GDS occurs actually above what we limit the fluorescent range to. So if you look at a textbook, with GDS, this is really the only portion you see is the exponential phase. You do not see the plateau phase, and you'll notice that in all of your graphs. The only other thing I really wanted to go through right now, and with this interpretation guide, is the internal control. The internal control, the lines are very similar to positive sample lines in the test window for the actual positive result. There is a threshold line, but the main thing to remember about a negative control that is a little bit different is negative controls are there to validate, internal controls are there to validate negative samples. If the target line for an individual sample stays below the threshold line, the internal control for that corresponding sample must cross the threshold to be a valid result. If the target line stays below the threshold, and the internal control line stays below the threshold, then you get our third result, which is a no amp, which means no amplification. That sample needs to be reanalyzed from the beginning, go back through the sample prep process, and then reanalyzed on the instrument. So that's the main thing about internal controls, is they are there to only validate negative samples. And we'll go over a run file here in a minute that shows a little bit more about what I'm saying there. So back to what really the main thing labs need to understand is this curve interpretation. It's just two files. In example A, this is what you're going to see significant portion of the time, very valid positive. If you remember from our guide, samples moving across horizontally. Here's our number of cycles. As you can see, this is around 12 cycles. The exponential growth in the line starts hitting. It doesn't go straight up, but it goes up at a very steep clip. It's an exponential growth in fluorescence. This one starts just right after 15. As you see, it crosses around 20. Extremely typical positives here, no question about these. The other graph on this page is really what we see. You will see these on a regular occurrence too. These are what we call more weak positives. Definitely still a valid positive. If you notice, the line is moving up in an exponential fashion, not quite at the slope that you see here when we have much more stronger positives, but still a very valid positive. The line is moving upwards. Those are the two things that everyone needs to be aware of and have full confidence, and that result is a valid result. There are no questions around those. Exponential growth in the curves, just one is much earlier. This indicates there was a higher cell count in the post-enrichment than this one, but still definitely typical amplification occurring. Anyone got any questions on that? That right there is really the main thing about graph interpretation on the positive side.
Just quick questions on them. I know most of the time we're seeing the line go below the zero value. Is that just part of the software, or what's going on there?
Yeah, that's perfectly normal. And you see in both of these examples that is the case. This one, both of them do that. That is just background fluorescence that occurs in the tube when it goes below the threshold, the algorithm, there's just very limited background fluorescence. You'll see a lot of times you'll see float in the lines, and that's just a little bit higher background fluorescence, but the algorithms in the software, they're just crunching that data, and those lines will move across horizontally. Either you see them above or below the line, either one's perfectly normal within our system.
And then also, you said sometimes you'll see float, and sometimes, how often does it refresh the data? Because your guys' platform, you can actually see as it goes. How often does that graph refresh?
That's a really good question, Kyle. I think it's every 3 minutes.
3 minutes, okay.
It's in that range. I think it's in between 2 and 3 minutes. But if you watch it, like you said, you see it's real-time analysis, right? As the DNA amplification occurs, when it gets down, when the temperature drops where the DNA comes together, if it is the target sequence, the probe will attach. And when it does that next reading, you see the jump in that line.
Yeah. And then I've seen it where sometimes it'll jump up and then the next cycle it kind of levels back out.
Correct.
What's going on in that?
That reading, there was a little bit higher fluorescence level. And that's why actually you do not see the first 5 cycles, because there's not enough data and the lines are moving all over the place. But as the instrument gains more data into the algorithm for the software, the lines will, what we call normal. If you look over here, it says normalized fluorescence, right? What we don't see is the raw data gathering that's actually occurring. And as that data comes in, if say that reading, the fluorescent level was just different than the previous ones, it can cause that line to move. But then the next reading, it levels it out, right? So it's just a little bit difference in the fluorescence reading and and that cycle that caused that line to move, float in a different fashion, and then as more data is collected and analyzed, that line will level out.
Thank you, Rodney.
You're welcome. So, um, like I said, I really wasn't going to get too much into this file. You guys are, I believe I've sent it to quite a few of you, but this file really goes through a lot of different things. This is the MPX top 7. This is I'd say our second most complicated assay. It's a 4-plex assay with an IC, but still the information is the same. The lines are moving up exponentially. This is what, Kyle, you were talking about. There's some pretty significant float in this line where these lines die below the threshold, or at least some of them are dead equal with it. But that is just normal background fluorescence. If you notice, there's no exponential growth. It kind of just meanders along, right? Yeah, it is floating upwards, but it's not an exponential growth where there's actually de-amplification occurring. Just another weak positive. I'll get into more of these invalid results in real GDS runs right now. So let's jump over to those. I've got a bunch of run files up. Can everybody see the run file I have up now?
Yes. Yeah.
Okay. So this is obviously an extremely typical positive, just like what we just saw in our example. Starts heading up around 11 cycles and then just moves up exponentially. As we talked about, you do not see any plateau phase that's occurring above this 0.2 level of fluorescence. So this is extremely valid, typical positive. This is what we just discussed. Float in the lines. This is perfectly normal. Some of them are down below the threshold here, below the 0 point. Others just float through here. This is an extremely normal run. ICs all look good. Very pretty much no inhibition. So sample prep was done great. None of the matrix was interfering with it. Like I said, this should be a high percentage of your positives. And if this isn't the high percentage of your positives, or the other percentage would be these a little bit weaker positive, but still an exponential growth in the graph. A lot of float going on in the lines here, perfectly normal. But if you notice, this line is moving up, not floating along. So these are our two valid positives that if the labs use that example with A and B, they should be able to see, well, definitely, all the valid positives. When any a different type of result that we're going to look at here in a minute, we'll look at a bunch of invalid results too, but we can look at like the next run, just multiple positives here, extremely valid, right? Just great amplification, just really no inhibition whatsoever. ICs look great. Minimum float in the lines here. This is just a perfect GDS real-time PCR analysis. Great sample prep. Just no questions there. Those are really the valid positives. There's not a whole lot of variation in them. Like I said, this is the only variation outside of these two that you'll see. Any questions around valid positives? If not, we will move to invalid positives. This right here is what I would say is going to be 95, 99 probably percent of invalid positives. What this is, and if you notice this line obviously touching the threshold, gave a positive result, because anytime it touches this threshold, it's going to give a positive result. But if you notice, the line is just going at a complete diagonal. It never does the turn up and does the exponential growth in the line. What causes this is background fluorescence that the instrument is reading that never essentially decreases, so the algorithm will crunch this line, for a lack of a better term, down below the threshold. There's just a lot of background, and we don't have great explanations for this because you'll reanalyze the sample, and that background fluorescence will not be there, and you'll get a valid result. But this is what you're going to see when the results are not valid positives. I would say almost exclusively outside of a couple others, and usually the others are going to be revolve around broken instruments, or all the contents evaporating out of the amplification tube. But this is your number one thing, and we go back, it looks nothing like our valid positives. But like I said, this right here is never a valid positive. There's, you can just compare it to the IC in the run that is obviously DNA amplification, which, mimics our very typical positives. Look at this. This is a great run. The ICs are showing no inhibition whatsoever. The other lines are essentially flat, but this one just, you can see it started floating at what, 2.5 cycles is where the reading, and then it didn't cross until 20 cycles. That's not normal at all. That is an invalid result, and that needs to be reanalyzed.
Hey, Rodney, on any
Yes.
given run, you can see a mixture of valid or invalid results at one time? Because I know we've seen that come back where we'll see wells 1, 2, and 5 are typical, well 5, well 7 is atypical invalid result. Is that common to see in a run?
I wouldn't say it's common, but it absolutely it can happen. Just positives in general are not very typical themselves, right? There's very low level positives, which obviously good for the customer, but so it doesn't have any bearing on any other positive or negative sample. Those are always isolated to those individual tubes when you get a result like this. If there was any other positives in the run, that those stand on their own, and just like these negatives do. And that's what I meant earlier when I said, we don't really have a great explanation for this, because you can reanalyze those samples, and that background fluorescence is not going to cause that to happen again. I don't believe I've ever seen a sample where you get an invalid positive result and then you rerun it and it also provides an invalid result. I've seen no amps, of course, occur on the same sample, but never have I seen invalid positives like this occur back-to-back runs on the exact same sample. So we look at some others. This is just another not valid positive. There's a lot of float in these lines. Obviously, the software, these lines were able to stay below the threshold, even though there's a lot of float. But as you notice, 2 of the lines did cross the threshold. But if you notice, once again, they started, their ascent upwards, very early. And these invalid results will do that. Where valid positives, they generally are going to be along the bottom and then they hook upwards, usually after 12 or so cycles. Every now and then we'll look at some very, very strong hot positives, but you see these start and then they just go up in an angle and then cross, usually 15 to 20 cycles after they start that initial jump. And, that's not normal. When we look at truly valid positives, you can see these started their upward movement. I don't know, was that 12 cycles? And then this cross, that's the green, cross within 3 cycles. Generally, I'd say they'll cross, if you notice where they start their initial increase in fluorescence, they'll cross with, I'd say, a significant portion of time within 6 cycles. And then some will take, maybe 7, probably at most 8. But those invalid results, they start up at around, this one started once again about 2. It took 18 cycles for that to hit the threshold. Not DNA amplification, right? Not that exponential growth. Um, sorry. Here's another example, just moving along at an angle. Um, not the typical DNA amplification. So once again, if we look at this started up, about 2 cycles, took it all the way to 16. So it took 14 cycles to cross the threshold, but never did increase in fluorescence. Just basically a slant, not the exponential growth like you see when you have normal DNA amplification. Okay, so here's another couple of positives. You look at this, nothing really valid about this. Line starting up and diving down straight, just not anything looks normal about these. This right here, if I was willing to bet, potentially cracked amp tube or lid wasn't closed properly, contents basically evaporated out of the tube. When that happens, the instrument is just reading, what's ever in the tube, and there's just not enough data around that to basically give us normalized fluorescence.
In an instance like this one, would you rerun everything, or would you just rerun the positives?
So if I were looking at this, if I were analyzing this, what I would say for sure, those two were not valid. I would probably have said rerun the entire run, and for multiple reasons. The controls, you can't say that that's a valid positive for your control either. So in a run like this, it would be best case to reanalyze the whole run, just because there's nothing good about this run.
Thank you.
Just another, going back, just wanted to throw in a weak positive here. Perfectly normal. So the reason I brought this up is if we talk about moving on to like internal controls. So internal controls, if you notice in your runs, they're by design start amplifying around 18 to 20 cycles. The reason why R&D designed the assay that way is we did not want the amplification to start occurring early on in the run, because remember earlier when I talked about plateauing of the lines, the reason that, and you remember the reason I said is because you run out of reagents. So we don't want the internal control, because the internal control, yes, it's a different segment of DNA, of course. It's just a little strand that we put in there to verify amplification. But it's using the same nucleotides to build the double-stranded DNA that the target is also using. So we don't want it to use all those nucleotides building a double-stranded DNA that, like I said, we can't see it, but it would, it's continuing up off this page. If it will run as long as it runs out of reagents. But if you notice, the earlier we have positives, the stronger the positives, the hotter the sample, the more it impacts the IC. Because basically it's doing exactly what I just told you we did not want the internal control to do to our DNA target. Since this started amplifying so early, it uses up the nucleotides. So the internal control for that corresponding sample does not have nucleotides to build its double strand. And this goes back to what I was talking about earlier. Internal controls are only to validate negative samples. So when we look at these samples here, all of these have to cross the threshold for these to be valid negatives, If any of these lines that correspond to these did not cross this threshold, we would get that no amp result, need to be reanalyzed. But when we look at the impact this has on the IC, stronger positive, you see how it affects the IC. The next strongest positive, this IC, and then last, this IC, with a very, I'd say it's minimum impact, but you can see it when you include it in with these, how it's starting to affect it when there's the samples are negative, so there's no DNA amplification occurring. And when there's a later positive, this is not late, but it's not as early as these, you see that had no impact on its IC because there was plenty of nucleotides to build the corresponding internal control for this sample. So this shows you how hot positives can impact the IC, but it's not going to affect the validity of the results. Now here is an extremely hot positive with a CT of 11. And if you notice, its IC had absolutely no reagent left to build its double-stranded DNA, but we still got a valid positive result. The reason why, remember what I said, ICs are there to validate negatives, and what they do is, they ensure that DNA amplification could or did occur, and had there been targeted DNA, it would have been able to amplify. Well, in this case, obviously there's targeted DNA because we got a very valid positive result in the positive window. So the IC actually no longer plays a role in determining the validity of the result. So you can see this on really hot positives. Usually the CT needs definitely be below 15 and usually closer to 10 cycles. And really anything below 10, you're going to really see a IC that's really has a very limited DNA amplification to it, if any. We don't always see it in E. coli. Obviously, you see it here due to the shortened enrichment time. Where you'll see something like this occur more often would be in the Listeria assay, and that's due to the longer incubation time. If a sample has a decent number of starting Listeria cells at the beginning of the enrichment, with the enrichment time being, definitely in the 30 plus 24 hours, you're going to see a lot higher cell counts post-enrichment. So you don't need many cycles for that level of fluorescence for that probe to attach for it to cross the threshold. That's where we see more impact on the IC with positives is in the Listeria assay, but you can see them in any assay. The enrichment time is just so much shorter for E. coli. We don't usually get to those cell counts. Does that make sense?
So with that, Rodney, so you're saying we can have valid positives without it being above the threshold on the internal control, basically.
Correct. As long as the target window is valid, which, what we went over earlier, this is a valid. Remember, this only validates negatives. So if this window is valid, you can see this. Like I say, you really don't see it often in E. coli, Salmonella. Listeria is where you'll see it most, but when the cycle, like I said, is around 10 plus or minus, you can see it. Sometimes I've seen it happen when it's close to 15, but that's pretty rare. Okay. So if we look at ICs some more, when we look at this here, if we remember what we talked about, ICs do not start amping until 18 to 20 cycles. They have the normal DNA amplification, which is exponential growth in the line. And if we isolate this sample, there's no positive control that's going to start amping at 4, cross the threshold at 6 or 7, and then dive back down. I'm pretty sure this is another case of the contents evaporating from the tube. So even though we had a negative result here, and that's due to this line crossing the threshold, this is not a negative result. This sample needs to be reanalyzed. But once again, we fall back on our knowledge of what DNA amplification looks like, and that is this. So if we look at this, we have completely negative in the target window. The internal control stayed fairly flat, gave us a no amp result. This sample needs to be reanalyzed along with this invalid result here, due to most likely contents evaporating from the tube. Any questions on invalid IC? Like I said, it really follows the same logic as what you look at in your target. You can see inhibition in the IC, which will essentially give you more of what we would, in comparison, think of like late positives, where they do amp, but they cross the threshold late. That's usually due to matrix inhibition. But those are still valid negative results, just like you have valid negative positives that cross late. So I did bring this MPX top 7 result up. Perfectly normal graph here. This is the same genetic target that's in the standalone E. coli 0157 kit. This is universal EAE, and this is shigatoxin genes. This will tell you which shigatoxin gene it is. Is it shigatoxin 1 or 2? Our ICs in the small window down here. As I mentioned earlier, no one needs to understand what combination of genes causes a positive result. The software will calculate that for you. The only thing that needs to be done is verifying are the lines valid using this principles that we've discussed previously. Any questions on that? I guess, one thing I did not pull up, I think this assay right here is going to be MPXID. MPXID, that is the only two-tube assay we have due to the number of gene targets we're amplifying. And the software will aggregate those 2 different amp tubes to provide you a single result. I hope this is in PXID. I think my computer may be out of memory. While this is spinning, anybody have any questions on what we went over so far? Sorry, everybody. I have faith it's going to open up. If it doesn't open up here in 1 second, I'll see if I can close. Oh, here we go. Yeah. So MPXID, just the one thing to make sure everybody's aware of is, there's 2 groups of amp tubes in the test kit, group 1 stack, group 2 stack, and they need to be put in the software in the appropriate order. As you see, this sample right here, sample 1, it corresponds to sample 5, the blank 6, positive control 7, and then this sample here corresponds to 8. They do need to match like that. And when you look at the results, it's only going to show you the group 1 and at a time, but really what the lab needs to do is look at the combined results. They don't really they need to look at here and make sure everything looks normal, but they don't need to try to understand what causes which organism to be positive. The system will combine, tabulate the results, and if you see this sample number here was positive for O103. So definitely no need to try to understand any of the combination of genes. That was all of the different types of runs I have. Really, it goes back to understanding that there's really 2 lines that everyone needs to know that are valid positives. Pretty much anything outside of that, needs to be questioned. And most likely the invalid positive is going to be that line that basically grows at a slant. That's what they're going to see as far as invalid results almost all the time, except, like I said, the other examples are broken instrument or no contents in the amplification tube. Any questions on any of those run files or anything I didn't go over?
You did bring up one graph that did have some fanning on the IC side. Should there be any concerns when we see fans like that for negative samples? I know you talked about for positive samples, it'll kind of plateau and be shorter, but what about for negative samples?
No, you guys, a lot of the samples you're testing are, beef samples, and what that is just a little bit of inhibition from sample matrix on DNA amplification. You see it probably more often in ground beef samples than you do trim, just due to the levels of fat floating in the enrichment. What I would say, I wouldn't worry about fanning of ICs for internal for the ICs unless you're seeing it all the time. If you're seeing it all the time, you probably want to understand why that's occurring. Is the sample being drawn out of the bag? Is too much sample matrix being collected in the 1 mil? Is there the full wash procedure not being conducted through the full 10 seconds? Is it a little bit inadequate volumes? So those are things that can cause fanning that if you were to see it on a regular basis, and the reason why you really want to know that is because if you're seeing that, what that's going to lead to is no amps. And then if you see that, you can head that off and try to get that corrected before you start getting no amps and then having to reanalyze samples. That's really the concern, is where that leads, not as much at all really the validity of that result.
Understood. Thank you.
How does everybody feel? Do you feel better about what graphs look like, or any reasons behind it? I could definitely go into the reasons why certain things happen, but I think I covered the few that you're going to see most time. Some of really odd looking graphs. There's different reasons. If you want to look at that that one interpretation guide, it has more graphs with a little few lines as to what we believe caused it. And we're pretty accurate because we worked with the lab and understood exactly what happened. Like I said, if you want to know what happened in a run, we can look at it. We can't always tell you, but we use historical knowledge to give some of them.